So good morning, everybody. Thank you for that, Larry. What a fantastic start to an amazing symposium. My name's Simon Lovestone. I'm from London, UK. So it's just great to be here, and what a wonderful symposium it looks like it's going to be, and it certainly was for the first one that I came to last year. I just want to say a couple of words of thank you to Larry. I think this is an amazingly generous symposium. I mean, generous in lots of obvious ways, financial ways, I imagine, but I really mean intellectually generous because of the scope of the subjects that we'll be getting really into over the next 2 days. It's also generous in terms of time. You know, the speakers have been given a reasonable amount of time so that we can really get into these the subjects in real detail. And I was thinking about this as I was coming over, and I think these symposia, they're a little bit like TED conferences, but they're like TED conferences for people that still have an attention span. kind of like they're kind of like TED for grown-ups. Yeah, I think that's that. So I'm moderating this session, and I can't remember what it's called, but I think it's more or less Little Bugs and How to Kill Them. And, you know, I don't know whether you know this, Larry, but I think Larry's played a really cruel trick on me in asking me to moderate this first symposium, because I started off life wanting to be a microbiologist. So my early career was in microbiology. I really, really wanted to be a virologist, and I got waylaid partly by the amazing discovery that proteins can be infectious agents, and partly by the much younger and more attractive wife of my teacher, who really didn't have a large nose and who I still have some apologies to make to, but maybe that's a story for another day. I'm a wannabe microbiologist. I've always wanted to be a microbiologist, really, and I look at this field with you know, real interest, and I'm really looking forward to the speakers. Like some people want to be rock stars or want to be sportsmen or women. I'm a wannabe microbiologist. And so I really, really like the topics that the speakers have got, and they've been given 45 minutes. We're going to do 35 talking and then 10 minutes. But, you know, don't worry about the time, chaps. I like this subject so much that if you want to take 90 minutes each, that's good for me. And that was your only error of the symposium, giving me this moderating. So we're going to start with Christopher Walsh. And he said, don't say anything about me, maybe just 6 words, because it's all in the program. So I'm just going to say 1 word. He's from Harvard. I am going to say, though, I looked on Wikipedia, and I didn't know this, Chris, but I see that Christopher Walsh has a first author paper in Nature with E.O. Wilson, who really, what an intellect that man. And I think any man that can have a Nature paper with a man that writes a perfectly passable novel about ants is good for me. So, Christopher Walsh.
I'm happy to be here at the invitation of this old guy, whom I met actually in the year of Super Bowl I. Larry was a postdoc when I was a graduate student in the Lippmann lab at Rockefeller. This is how I think about Larry. That's how he looked before he went white. I think I'm happy to be at this meeting largely because it's the first time my wife and I have ever spoken at the same meeting. So if you don't like what I have to say, she'll be much better. These are hieroglyphics that remind me to say I'm going to talk about antibacterial agents, not antivirals, not antiparasitics in this talk. And antibiotics are largely small molecules that kill bacteria in principle by selectively hitting bacterial targets, even though they're optimized relative to humans. Here's a slide from about a decade ago which said we should still worry about infectious disease on a global scale because in that year about 15 million people died, about 50% of them from bacterial diseases, about 50% from viral diseases. It's still a big problem globally. In fact, the message of this talk is in these 4 words. If you don't remember anything else, we have a lot of bad bugs and we have essentially no drugs to treat new life-threatening bacterial infections. Over here is a circle of the indications that the FDA will approve new antibiotics for. This is community-acquired pneumonia, urinary tract infections, which Larry likes to think about, and interabdominal infections. Any new antibiotic that comes is a result of clinical trials in one of those settings. Now, I live somewhere between the chemistry, biology, and medicine universes, but the presentation I'm going to give you today is chemocentric in terms of thinking about what we need to treat bacterial diseases. Now, I'm not a very good person. I don't do yoga. Unlike my wife, who seriously meditates, I don't know how to meditate. So instead, one day a week, I do biotechnology. We can debate the comparative therapeutic benefits. I'm still here, but barely. Now, I do a lot of biotechnology, as I said, and I'm happy to talk about the other 8 things at some point during the next 2 days, if you'd like. But I call out a company called Achaogen because that's about new antibiotics. And really, this is a statement that there are conflicts of interest. Anybody who's on the board of a company, you should be suspicious of what they have to say. I will not knowingly say anything about Achaogen, but nonetheless, you have to be suspicious that that's the context. Now, here are some of the players that are important human pathogens. The causative agent of tuberculosis, Mycobacterium tuberculosis, frequent pathogen in UTIs, and also in infections of the inner lining of the heart, Enterococcus faecalis, strep pneumoniae, and Staph aureus are 2 of the most famous organisms that cause pneumonia. Another culprit for UTI infections, and then Haemophilus influenza is a frequent pathogen of respiratory tract infections. Now, these 4 are so-called gram-positive bacteria, gram-positive, and these 2 are so-called gram-negative. And what is gram? Gram is a stain that was invented by a microbiologist in the 19th century, a Danish microbiologist named Christian Gram. And things that stain positive have a different kind of cell wall. These are cocci, spherical, from the gram-negative rods of bacteria that you see here. And all that matters for this discussion is that gram-negative guys have a more complex cell wall. It's harder to get antibiotics into gram-negatives, and so in general they're more resistant, intrinsically more resistant, perhaps more optimized in Larry's terminology of evolution. So 4 gram-positive pathogens, 2 gram-negatives that are serious problems. So the title of this lecture was sort of past, present, and future. Where have we been? Well, one could say the birth of the antibiotic era started in the 1860s with Pasteur and his belief in and proof about the germ theory of disease, of course, involved in the first vaccines. Robert Koch, who invented a set of postulates which allowed one to identify causative bacterial pathogens and to identify them as the cause of disease. And then a man named Ehrlich, who is synonymous with the phrase magic bullet, as the first person who came up with synthetic molecules that would cure bacterial infections. And I would argue over the past 100 years, there have been 2 parallel and independent lines of discovery that have been productive and fruitful for finding antibiotics. One is to look in nature, in a sense to discover the antimicrobial weaponry that bacteria use in the warfare in the soil against each other. And then a separate line, this sort of Ehrlich approach that chemists can devise magic bullets that will kill bacteria. And indeed, if one would go down the list of perhaps the most famous of the antibiotics, penicillins, erythromycin, tetracycline, streptomycin, vancomycin, probably in your lifetimes you've had most of them in one bacterial infection or another. These come from nature, from bacteria and fungi. And then here's ciprofloxacin, which was the anthrax scare drug that people were hoarding. Sulfa drugs, they're man-made. The most famous of the microbiologists, Simon, the person you wanted to be, was Alexander Fleming, of course, for finding the most famous of the antibiotics. Here's the beta-lactam warhead of penicillins, called penicillins because they were isolated from a mold or fungus called penicillium. On the other hand, here again is a picture of Ehrlich, who was really involved in the synthetic dye chemistry in Germany, and they made a lot of colored molecules and tested some of them for curing infections. And it turned out that a sulfur drug, here's the sulfur in yellow, were active as antibiotics. This molecule is called Prontosil. It was the first one commercially introduced as a magic bullet in the 30s. And amazingly enough, 80 years later, the sulfur drugs are still being used as antibiotics. Sort of spectacular accomplishment. There's Fleming again, and here are these two guys who during World War II were able to convert his laboratory discovery in the late 20s into full-scale production of penicillin as a life-saving antibiotic in the second half of World War II. Waxman won a Nobel Prize for discovery of streptomycin, and this sort of unleashed a golden age, as it's called, of antibiotic discovery, which lasted about 20 years, from 1940 to 1960, and all of the antibiotics a few of you may ever have heard of, isolated from nature, were found in that 20-year period. Here's the Prontosil, discovered in the 1930s. And in the intervening 50, 60 years now, from 1960 to today, there have been essentially no new molecules discovered from nature that are broad-spectrum antibiotics. And instead, I would say it's been a golden age of medicinal chemistry, in which chemists have tinkered with the periphery, leaving the warheads of these molecules intact and fighting wave after wave of resistant bacteria, which emerge whenever you use an antibiotic widely. So we're perhaps at the end of that golden age. It turns out, compared to, let's say, cancer, where there appear to be hundreds of relevant therapeutic targets, antibiotics and the treatment of bacterial infections have historically been a target-poor therapeutic area. Even though there are thousands of molecules one can isolate that at the beginning look to be promising antibiotics, in fact, only 5 targets are commercially, medically, and therapeutically viable. The inhibition of cell wall synthesis in the bacteria, the selective blockade of bacterial protein synthesis, but not human protein synthesis, interdiction of the information transfer from DNA and RNA, the inhibition of the synthesis of a vitamin called folate, and then disruption of cell membranes. So let me just briefly walk us through those 4, and to say the blockade of the production of the cell wall in bacteria is where the penicillins act, the most famous of the antibiotics. Interdiction of protein synthesis in bacteria, streptomycin, kanamycin, erythromycins work there. Blockade of DNA to RNA to proteins, ciprofloxacin is a representative fluoroquinoline, works here, and the frontline drug for the treatment of tuberculosis and drug combinations, rifampins, block RNA polymerase conversion of RNA into proteins. And finally, it is the provision of the thymidylate monomer for DNA synthesis, here's a jargon, excuse me, that its biosynthesis is where the most ancient of the synthetic antibacterials, the one in 80 years of continuous use, the sulfur drugs work. And that's it. No other targets. So where are we now? Well, I'd say we have in our armamentarium, when you go see an infectious disease or primary care physician, he or she will give you a drug which works against one or more of those target classes. Unfortunately, as you can read in the literature and papers, multi-drug resistant bacteria on the rise. Now, in the past decade, genomics has resulted in the sequencing of maybe 5,000 bacterial genomes, including all of the major pathogens. And so you say, okay, with all of that genomic information, we should be in a much better situation. And unfortunately, 15 years later, we have not a single new antibiotic which has arisen from that bacterial genomics database. So here's the problem that we face at the moment, and it's the sort of yin-yang of whenever one utilizes or deploys a new antibiotic, antibiotic resistance occurs. Larry, in his inimitable way, reminded us about mutation rates, and any antibiotic once deployed will kill the sensitive bacteria, let's say 999,999 of a million, and the one resistant one will live and be selected for. Now all the others are dead. It can feed off of those dying bacteria, and you'll select for the growth of a resistant organism. Since you and I may have 10 to the 14th bacteria inside of us, and they, in fact, in our intestine multiply every 30 minutes, resistance is inevitable. As sure as night follows day, when we deploy a new antibiotic clinically, the clock ticks down on its period of use. Here's a table from an article about a decade ago that says, pick whatever antibiotic you've ever heard of. Here's the year it was deployed. Here's when clinically significant resistance was observed. Methicillin-resistant Staph aureus. Those are the flesh-eating bacteria that you read about. Antibiotic introduced widely clinically 1 year later, significant resistance. Bacteria are smarter than Larry and me and probably the rest of you. Here's an experiment that was done by a former trainee of mine, Jerry Wright, who went outside of his laboratory in Toronto and picked soil from the bacteria, and he selected bacteria that form spores, actinomycetes. They're easy to isolate. And he said, how many of them are resistant to a panel of 21 widely used antibiotics? And amazingly, the average was 7. And some of them are resistant to 12 antibiotics, which suggests that all over the world, antibiotic resistance is already there, waiting to be tapped. And so, Larry, what would a conference at the GoldLab be without an omic? Here's an omic. It's the antibiotic resistome. So where do antibiotic resistance genes come from? Of course, they're in drug-resistant pathogens. They're also in the guys who make the antibiotics in the first place. You can't be a penicillium mold making penicillin unless you have a mechanism for self-resistance. They're also in other bacteria. They're not turned on. And they're even in precursor genes, housekeeping proteins. So anytime we discover a new antibiotic, natural or even synthetic, the resistome is there waiting to respond. I think that's one manifestation that I would say when Larry says bacteria are optimized, they're optimized to play defense. So going forward, here's a quote from the New York Times. It says, resistant superbugs threaten the actual treatment of infectious disease. Here's the other 4-letter message. I'm not sure that I believe that, but this is about the problem of antibiotic resistance. In fact, I would argue it's a tragedy of the commons in a way that very few other drug resistance problems are. You have an individual who's resistant to cancer drugs. That's not an infectious spread. But antibiotics are sort of the only drugs taken according to the prescribing physician. When you take them, you put everybody else at risk. So here's a sort of quantitation of it. Over the last half century, it's estimated that 50 million tons of antibiotics have been prescribed, dispensed, taken, and peed out. into the environment, selecting for resistant bacteria, so that my effluent is a problem for the neighbor downstream. It's led to the global spread of multi-drug resistant pathogens, and here's the sort of death toll in 2010 resulting from acquired bacterial infections by drug-resistant bacteria. I had a 5-way bypass about 11 years ago, cardiac bypass, and the thing I was most worried about when I woke up was, could I get out of the cardiac intensive care unit without a vancomycin-resistant enterococcal infection? I did, but 6 of the other 8 patients in that ward did get VRE. So here's the chief medical officer from the land of the frozen queen. In January 2013, here's a quote, but she argues that antibiotic resistance is now so serious a threat that it should be added to the government's list of current civil emergencies. The sort of code red every day you get up in London, you should think, oh, the bugs could get me today. Boulder, maybe it's better here, I don't know. In the spirit of David Letterman, here's a top 10 facts list, not produced by TV guys, but by the European Center for Disease Prevention. And these are my bold, that antibiotics carry the seeds of their own destruction. That means when you use them, they select for resistant bacteria, so their lifetime drops. The pipeline is running dry, and I'll turn to that in a moment. We're certainly using up our reserves, and all of us talking about imperfect sociology have to practice antibiotic stewardship, but when your kid or grandkid has got a serious infection, you want antibiotics, even if they tell you, oh, let's wait another 3 or 4 days, maybe it's viral. You say, yeah, I understand, but I'll go to another doctor if you won't give me antibiotics. So hand in hand with better antibiotic stewardship, I say there's a pressing need for the next generation of antibiotics. As soon as we use the current ones, the clock ticks down, and where will they come from? Well, one of the problems, I would argue, is there has been a 40-year innovation gap from 1960, when the quinolones were introduced, into 2000. There are lots of reasons. One of them was complacency on the part of the public and research. A US Surgeon General famously said in 1965, we have conquered bacterial diseases. We don't need any new antibiotics. It's important to understand biology. Now, this is just one version of a flowchart of drug discovery that says to find a new antibiotic, it might cost as much as $1 billion before you're done. Most pharmaceutical companies don't have the appetite to carry out that research anymore, because the average amount of time an American takes antibiotics is only 14 days a year. The average time, Viagra, 250 days a year. Much better economic argument. I don't take Viagra at all. Not good for my heart. How will we generate effective new molecules? There are a bunch of other things on this slide, but let me just sort of take up that one. Here's a pictogram again of beta-lactams and the quinolones and the erythromycins, the tetracyclines, first generation all the way out to 4th and 5th generation. And one of the questions is, we've lived for 60 years finding one generation after another by tinkering with the periphery, leaving the warheads intact. What's the chance that's still going to be fruitful in the future? I think low. Much more useful, I think, would be finding new scaffolds from nature, and I don't intend that anybody recognize any of these, but the name here is kind of cute, abyssamycin. This was found from a marine bacterium 7,000 meters down in the Marianas Trench from the abyss. And I think it's likely that every time you look at a new biological niche, you find new bacteria, perhaps new molecules, and maybe that's the way we have to go forward. So let me leave you with 2 ideas. Unlike oncology and HIV, where combination therapy is the norm, historically, in treatment of antibiotics, monotherapy has been what people do. But nobody these days would treat an HIV patient with a single drug or a TB patient. So I think it's important to change the view of physicians and researchers that will need to go away from monotherapy and into the modern era of combination antibiotics. And also, we'll be able to identify bacteria with the kind of technology Sociological would use, and then we could use narrow spectrum antibiotics rather than broad spectrum, and maybe we'd have a better yield. So the abandonment of the infectious disease area over a 10-year, 15-year period by most of the large pharmaceutical companies has created a vacuum of discovery and innovation, but I could argue it's an opportunity for biotechnology companies and academic discovery efforts. And here are 6 small companies that have formed over the past decade or so. And in fact, the best-selling recently approved antibiotic is daptomycin from a small company called Cubist. Here's the Achaogen molecule where I'm on the board. So I do think this may be, in fact, with government and other assistance, an opportunity to fill in the void. Summary thoughts. The drug pipeline is substantially unfilled. There's a dramatic need for new therapeutic strategies. And the message, bad bugs, no drugs. Hospitals are the most dangerous places you can be when an infectious disease specialist shows up for a consult. Thank you very much.
Plenty of time for questions.
Why don't I let you direct?
Thank you very much for your presentation. Not to spin a negative light on pharmacology, I recently got an infection on my arm and had to use a broad spectrum antibiotic to kill it. But is there a way that we might use more integrative or holistic type of modalities to treat the ever-growing problem of antibiotic-resistant pathogens, such as the Rife technology that was developed, I guess, many years ago, as far as all living things have electromagnetic signature. If you disrupt that signature, you can kill or weaken that particular life form or pathogen, as the case may be. So is the future ultimately going to have to head that way? I mean, you said that these bacteria are smarter than us, but Dr. Rife proved 50, 60, 70 years ago that, in fact, humans are smarter than bacteria. There's just a lot of money to be made in giving someone a drug versus giving someone a certain frequency that's going to kill a pathogen. So just any info on that. Thank you.
So if I understand your multifaceted question, one is, are there alternatives to antibiotic therapy, or classic antibiotic therapy? And I think the answer I would give you back is that companies were historically interested in broad spectrum, and you can talk about hammers, sledgehammers against bacteria, because in the absence of diagnosis of a causative pathogen, you wanted to cover your bases as much as possible. I think the problems with broad spectrum hammers have shown up. Patients get all kinds of colitis and other problems from getting rid of the good bacteria as well as the bad bacteria. So I think you'd like to have a lighter footprint in your therapeutic approach. One way of thinking about that is a narrow spectrum antibiotic. That means real-time diagnosis, which one can do these days, and have available specific bacteria which leave your commensals and good guys alone, and therefore is a much lighter touch. There hasn't been a commercial market for that. I think so large pharma wouldn't play that way, but $100 million to a small biotech company, $100 million a year, would be a reasonable return. It's so far below threshold for large pharma that it wouldn't work. Now then there are the questions of what about not using antibiotics, and are there other ways? And I think I would leave that to others, but if there's a fulminant bacterial infection, you've got 10 to the 11th bugs growing in some site on your elbow or your calf muscle, I think it's probably too late for that. So I think it's too late if you have fulminant infections. But the whole question of non-pharmaceutical approaches to bacterial diseases, less clear to me how to do that in an acute setting.
So I wanted to ask you if there's been any ideas or any push to design inactivating antibiotics, molecules that could, become inactive in the kidney or maybe in the urine. We know, that, for example, that some hormones, when taken, can become inactive after action in the body. So can we, by those means, even perhaps in the molecule itself or with a companion molecule, prevent diminish the dampening of the antibiotic in sewage and the environment? Has there been any progress in that?
So if I understand, there may be two facets to that question. The first is, does one ever administer a companion molecule with an antibiotic to prevent its breakdown? And certainly, in fact, the first carbapenems sold for years by Merck were a combination of the antibiotic thionomycin and a blockade of a renal dipeptidase enzyme, which would, during host metabolism, destroy the antibiotic. So that was a combination to make sure that the antibiotic lasted long enough in the host. I think you're asking a different question, which is, can one be sure that the things you pee out are no longer active and fully metabolized? And I confess I don't know anybody who's thought about a timing for inactivation so that when it's back in the environment, it's not active. It's an interesting question. I don't know how to do that, but I haven't thought about it. It's a good reason to have come to this conference. Yeah.
So first a correction, the Surgeon General never made that quote. That's an urban myth.
Okay.
The president of the American Medical Colleges did say in 1969 there's no longer a need to train infectious disease specialists as they'll have nothing to do but culture each other, but that's as close as...
Thank you. I like that one better, actually.
Yeah. So
I don't think the Surgeon General typically is somebody one should pay attention to.
Yeah. So since this is a symposium about biology and sociology, we spend about $10 in research for every cancer patient who dies as opposed to every infectious disease patient who dies. So we spend about $10 in research on cancer for every cancer death versus every dollar we spend in research on infectious disease. Yeah, low by a factor of 10. And you never see a Lifetime miniseries about patients heroically dying and battling MRSA or anything like that, but we do about cancer. And so I wonder if you could say a few words about why you think it is we're so much more frightened of cancer than we are of infectious diseases.
Well, I think we certainly see lots of things about HIV. Yeah, but because now HIV is a disease you can live with. I guess there's still the belief that bacterial infections are treatable. And if they're not, it's a failure of the system. But it's not the fear that if you get diagnosed with a bacterial infection, that's a death sentence. And so I think the sociology of fear is very different in the two cases. I told you I wanted to get out of that cardiology recovery unit as fast as possible, so there could be different microenvironments. But I don't think that's a good family TV story.
Hello. I'm reading an interesting book now, and actually it brings into question some of the lifestyles here that we appreciate in Boulder. But my question is about the prodigious amounts of antibiotics that are used to keep feed animals alive in feedlots, and how that is affecting drug resistance, and how we should go about maybe regulating that.
So this is about whether one should forbid the use of antibiotics as growth promotants, if I could put it as broadly as possible. And a number of European European countries, Denmark among them, early on forbade the use of antibiotics as growth promotants. So I'm very far on the side of saying that we should hold antibiotics in reserve for humans, not for making fatter chickens or heavier pigs. And there's absolutely no doubt that multi-drug resistant bacteria have moved from those animals into humans. So I think it's only an economic argument on the part of the industries, not a medically defensible argument. There's been over a 25-year period some motion to restrict, but bills come up every couple of years in the Congress and get defeated by the lobbying groups. So it's certainly something, if you want to have a conversation going forward, that would be a simple, clear thing to talk about.
I have, can you hear me? I have more of a basic question. You touched base on antibiotic resistance in terms of things that we give off, say, through urine or whatever, but not as much on antibiotic resistance with incorrectly taking antibiotics and then transmitting that pathogen that's now antibiotic resistance. Do you think that resistance stems more from the incorrect use of antibiotics or overuse, or from this excretion from us?
Well, it's hard to know what fraction of prescriptions written for antibiotics are unneeded, but there have been estimates that anywhere from a third to a half of antibiotic prescriptions are for illnesses that are not bacterial in origin. And that results from the slow time for diagnosis. It used to be, would take 2 or 3 days to get a culture. Suppose it were a third, then that's certainly a huge problem in terms of putting antibiotics into the local environment. But I pointed out, and so I think stewardship is really an important issue for dealing with outbreaks, epidemics in hospital settings, and in some communities where drug-resistant pneumonia causing bacteria spike. But I pointed out to you that even for a synthetic chemically derived antibiotic that presumably doesn't exist in nature, within a couple of years you see resistant bacteria with clinically significant resistance, which tells us that, to go back to Larry's point, they've had so many chances to optimize their responses that they will survive any challenge. Certainly, from a sociologic perspective, cutting down on the number of inappropriate prescriptions would be important. Second is completing a course of action. You feel better 3 days later, you don't take that 4th and 5th dose. So you've given a subtherapeutic dose, and that's the perfect situation for engendering resistant bacteria.
There's lots of questions coming. I think we've just got time for just 2 more. So up here, and then yourself.
It's my understanding that if you had been in a hospital in Scandinavia, you might not have been at risk for as much MSRI infection because they have legislated to reduce the amount of antibiotics used, that their hospitals are not as clean as ours, but they have fewer of these kinds of infections, so the rate has gone down. As a related part of this, you probably know about C. diff colitis.
I certainly do.
One of the most deadly kinds of intestinal and awful infections someone can have. the cure is not vancomycin for life. Well, that kind of works, but it's an awful solution. But instead, the fecal stool transplant. And that seems to be far more effective. Do you want to comment on this new side of antibiotics, which is looking at the way that the microbial community altogether can protect somebody against some terrible infections?
Yeah, so the C. diff colitis, that's Clostridium difficile, arises when you take large doses of antibiotics, you wipe out all the bugs in your GI tract while you're doing everything else, and the guys that can overgrow are spores of Clostridium, and that's life-threatening disease. So that's a good reason to go for narrow-spectrum antibiotics. In fact, there's been a recent approval of a drug called fidaxomicin from a small biotech, not from a large pharma company, to treat moderate cases of C. diff colitis. You've pointed out that fecal transplants have been used when they're life-threatening and nothing will work against those C. difficile infections. And the presumption is you're reconstituting the microbial community, and they are your best defense against the outgrowth of Clostridium difficile. I don't know whether it will become frontline treatment. I doubt it. I think one will only use it against patients who are in bad shape. But it's a reminder of how important the microbiome, the gut microbiome, is to our health. And one could have given a different lecture about the endocrine systems and the immune responses which are helped by a healthy microbiome. So it's certainly, as a reminder, there are 1,000 more bacterial cells in us than human cells. We are a composite mega-organism. We really need to respect not wiping out the commensal bacteria. Maybe one more question if we have time, Simon.
Yes, thank you very much. We've talked about taking known pathogens, organisms, and targeting them. But one of the huge issues out there is the unknown organisms. That is our ability now to begin to understand the role of microbes in disease and identify them and do so reliably. So, we have the old example of Heliobacter, but more recently, and Larry's aware of this, is a paper out of Denmark that talks about bacterial infections and chronic lower back pain, very severe chronic lower back pain, and really interesting results. And one of the difficulties there is how do we identify that the organism is in fact there? And I'm just wondering if you have any comments about that side of the equation, which is how do we actually identify these things?
So I think the question, if I could paraphrase it, was that there are certainly infections or pathologies where it now appears bacteria are involved. The classic case being Helicobacter and the causation of ulcers, but others are coming up. And so how does one use Koch's postulates in 150 years later to find the organisms? And I think the best answer one would give is to do multiplex genome sequencing, especially for organisms you can't cultivate in that particular environment. Try to sort them out. You may then, from a particular set of patients, come up with DNA which is only found in them as a signature of it. You might then be incented to culture looking for those particular bacteria. Then you have to determine whether they're the pathogens. So I think the identification of unusual bacterial DNA signatures in clinical samples would be possible, but it's only the start of sorting out which of those are actual pathogens, how do they cause disease, before you could think about treating. Thanks very much.
Thank you.